Amplicon sequencing has two important analysis steps: sequence clustering and taxonomic assignment. Before the NGS, Amplicon sequencing analysis conduct multiple alignments, calculating distance between sequence and estimating phylogenetic tree. After the NGS, because the number of the leads per sample becomes huge, sequence clustering with a threshold like 97% identity by conducting pairs sequence comparison becomes a major analysis strategy. And we call that strategy as OTU clustering. But OTU clustering have a problem. For example, in a case of OTU clustering with the sequence identity threshold 97%, you can find ambiguously assigned sequences in multiple OTUs, like blue one. These ambiguously assigned sequences are problematic for OTU clustering result because they can belong more than two different OTUs. To avoid the ambiguity, a method called denoising and dereplication has been rapidly used in microbiome research field. In this method, sequence errors are removed by denoising based on the number of sequences and their distances from a larger number of relative sequences. Then, completely identical sequence clusters called ASVs are used for analysis instead of OTUs, because OTUs conduct several thresholds like 97% sequence clustering, but ASVs are only used completely identical sequence clusters. So these are very different. And the idea is to remove the ambiguity of sequence clustering by doing denoising before complete match-based sequence clustering. Actually, OTU is a more general word, but in microbiome community analysis, OTU and ASV are obviously different concepts. And currently, most of the researchers are using the ASV method. And after creating OTUs or ASVs, in order to estimate the taxonomy of each sequence cluster, researchers perform a sequence similarity search. Although there are several reference sequence databases of ribosomal RNA genes exist, such as silver, green genes, and RDP, the majority of researchers use silver because of frequent update of the database. Since each reference sequence database uses different taxonomic hierarchy, it is difficult to compare the taxonomic composition inferred from the different databases. So, at least researchers need to unify the reference database within the research project. It's important, otherwise we cannot compare the taxonomic composition between studies. I described the bioinformatics of Amplicon sequencing, but how you could conduct actual analysis? You can easily conduct bioinformatics analysis of 6S ribosomal RNA gene Amplicon sequencing data using QIIME2. QIIME2 is an open source analysis environment that is written by Python programming language. Using QIIME2, you can conduct this kind of complex analysis by only typing some commands in terminal. Data2 is another good analysis environment for Amplicon sequencing data. And this Data2 is written based on R programming environment. Both of the tools have good document and tutorial on the web. And using these kinds of analysis environments, you can easily conduct bioinformatics analysis of Amplicon sequencing data.